we present a novel biomimetic model of the human eye and an innovative neuromuscular oculomotor control framework based on deep learning. Our muscle-actuated biomechanical eye model is fully functional, synthesizing realistic eye movements with 3 rotational degrees of freedom along with pupil and focal accommodation. We begin by overviewing each of our model's biologically consistent biomechanical and optical components. 6 extraocular muscles actuate the eye, which is modeled as a rigid object of appropriate mass within the orbital cavity. The cornea refracts light entering the eye from the environment. The aqueous humor further refracts the incoming light. The iris sphincter muscle constricts and dilates the pupil. The ciliary muscle actuates the deformable lens, which further refracts incoming light rays to focus them onto the retina. This is called accommodation. The vitreous humor also refracts light within the ocular cavity. Finally, our biomimetic model of the retina includes 14,400 photoreceptors shown here as black dots in a biologically consistent foveated distribution. Each retinal photoreceptor computes the irradiance it receives using dynamic ray tracing. Multiple rays cast from each photoreceptor through the finite aperture pupil sample the 3D environment. The size of the pupil and the shape of the lens determine the number of rays and their directions. Our eye model outputs a vector of photoreceptor responses that we call the optic nerve vector, or ONV. We next overview the visual motor control system of our eye model. In the sensory subsystem, the retinal photoreceptors compute their RGB irradiance values through ray tracing. The ONV feeds a trained deep neural network, or DNN, that has learned foveation control. Its output drives a trained neuromuscular motor control, DNN, in the motor subsystem that innervates the 6 extraocular muscles to synthesize realistic eye movements. The optic nerve vector also feeds 2 trained shallow neural networks, or SNNs, that directly control the iris and lens. All 4 neural networks are trained offline using data synthesized by the eye model itself. Without muscle activation, the eye droops in gravity due to its mass distribution. Its online actions are controlled through appropriate muscle activation signals generated by its neuromuscular controller. We now demonstrate the online operation of the optical organs, starting with the iris. A target light source of variable intensity is placed in the visual field. The SNN-controlled iris sphincter constricts or dilates the pupil to regulate the overall irradiance on the photoreceptors. The iris accommodates to the visual environment and to the position of the light source. Here, a visual target is moved in depth along the optical axis while the ciliary muscle adjusts the lens deformation to maintain the focal point of the refracted rays on the retina. Muscle contraction causes the lens to thicken and accommodate close-range targets, while relaxation does the opposite. Although the lens occasionally loses focus, the ciliary muscle visual motor control SNN does a satisfactory job. We next demonstrate full visual motor control in a variety of simulations. In this saccadic eye movement simulation, the eye rotates to fixate a randomly repositioning visual target under changing illuminations. The orange trace illustrates the noise inherent to biological eye movements. In this smooth pursuit simulation, the visual target is continuously moved while the ambient illumination of the scene is varied. Although not explicitly trained on moving stimuli, our model maintains the target near the center of the fovea and tracks it in a natural manner. Thank you for watching.